Hello, everyone. Welcome to the Toronto Geometry Colloquium. This is a weekly web series all about geometry processing. And this colloquium aims at promoting young researchers and researchers from traditionally underrepresented communities. And every week, we'll have an opener talking about their cutting-edge research for 10 minutes, followed by the headliner giving us a keynote presentation.
Hello and welcome to this very special edition of the Toronto Geometry Colloquium. I think we have a very biology-oriented geometry talk today, and despite the similarity in both of their topics, I think we're going to get to experience how broad and wide this field really is, which is part of the reason why I love it. Our first speaker is going to be a PhD student from our very own University of Toronto. He works a lot on modeling geometry for artistic applications, and you might have seen some of his work on 3D printed stop motion for facial animation before. If you haven't, you should look up those results. They look really nice. And in his most recent work was published in the Transactions on Graphics last year, he introduces a new way of interacting intuitively with musculoskeletal anatomy, which sounds really, really cool. And so all that means is if you know it's super important to model the musculoskeletal anatomy of creatures or animals if you want to make them look realistic. Without further ado, please welcome Rinat Abdrashitov.
All right, hello everybody. Hopefully you can see my slides. If not, let me know. Okay. Yes, so hi, my name is Rinat, and today I'll be talking about interactive modeling of volumetric musculoskeletal anatomy. And this work has been done in collaboration with Sangbei Bang, David Levin, Karan Singh, and Alec Jacobson, and we are part of the University of Toronto. So animating physically realistic effects like muscle bulging, skin sliding, and wrinkles without an explicit musculoskeletal structure is very challenging, and it does require skilled and tedious manual effort in order to achieve very high quality results. However, a user-friendly solution to the problem of creating a musculoskeletal structure that is suitable for character modeling and animation has been relatively unexplored. And the most common approach used in the industry relies on using sculpting software like ZBrush to model every single muscle. However, this approach is time-consuming and mostly out of reach for users who are not familiar with sculpting software like riggers, animators, and researchers. Alternative approaches like MyMuscle require tedious parameter tweaking and have a quite complex user interface. And these methods make it hard to produce intersection-free muscle shapes. If muscle shapes do intersect between each other or with the bone geometry, then that could potentially cause issues during the simulation. Additionally, often artists would load the skin mesh and need to sculpt the muscles to conform to the skin layer, which requires a lot of edits. Fiber directions play an important role in simulation by defining the directions of the muscle in which muscles contract. And fibers tend to point in the same directions as the gradients of hypothetical flow from one end to the other end of the muscle. And currently, artists need to manually mark the end of the muscles to approximate these fiber fields. So because of all these challenges that I listed, making incremental changes using all of these previous approaches is tedious and hinders the fast exploration of character design. So in our work, we take a very different approach. And unlike previous methods, we do not model individual muscle shapes as geometric primitives, so like polygonal meshes or NURBS. So we instead adopt a volumetric segmentation approach where every point in our volume is assigned to a muscle, fat, or bone tissue. And we provide an interactive modeling tool where the user controls the segmentation via muscle curves, and we visualize the muscle shapes using volumetric rendering. Muscle curves enable intuitive yet powerful control over the muscle shapes. So this representation allows us to automatically handle intersections between different tissues, like between the muscles and between muscles and bones, between muscles and skin. And it mostly automates computation of muscle fiber fields. Based on this volumetric muscle representation, we build tools that greatly simplify the creation of musculoskeletal geometry and can be used by non-modelers. First, the user starts by loading a skin and a skeleton mesh, or if the skeleton mesh is not available, we provide a tool to quickly generate the skeleton. It uses a pre-existing library of bones that are adjusted to fit the skin mesh. The user then generates a network of muscle curves that segment the volume into different tissues. And finally, we developed an algorithm to convert our volumetric segmentation into tetrahedral meshes that can be used for downstream tasks like rendering, for example. So first, I don't have time to go into details about our skeleton tool, but here's just a short clip that hopefully gives you an idea what it looks like and what it does. And it shows how we can use an existing library of bones to quickly and interactively generate the skeleton. Please see the paper for more details on this. Here's a clip of our muscle builder tool. So to create a muscle, the user draws a curve. Skeletal muscles require an origin and insertion points where the muscle tendons are being attached to the bone. So in our interface, we expect the user to always begin and end the stroke over the bone surface. And the first and last points of the curve are automatically projected via raycasting onto the surface of the bone to find their corresponding 3D coordinates. So then the depth values for all the points drawn in between is ambiguous. So we simply just linearly interpolate the depths of the first and the last points. And because we want the curves to be easily editable, we fit a Catmull-Rom spline in a least squares manner with 4 control points by default. So we have 1 control point for each of the end points and 2 along the curve somewhere. And you can see me kind of dragging and selecting those control points in the video. And the user can also easily add and remove more control points if they need to. So control points is how the user is able to change the shape of the muscle. So you've seen me in the video, I can move them around, but I can also specify so-called tissue values at each of the control points, which also affect the final shape. And we will explain in the future slides how those tissue values are used. So now, given that we've created this muscle network of curves and tissues values along those curves, we can generate, how can we generate the final muscle shapes? Well, first, let's look at this problem in 2D to make it easier to visualize. Let Amiga denote our 2D domain defined by a circle, which we can think of as our skin mesh. And we also given 2 muscle curves within the domain. Our goal is to find a scalar muscle functions for each muscle curve that describe the likelihood of any point in our domain to belong to that muscle. So take the first muscle curve as an example. We define some tissue values at each point of the curve, and now we want to smoothly diffuse those values away from the curve such that they become 0 on the skin boundary and 0 on the points of all the other muscle curves. And the same idea for the second muscle curve. And then similarly, we define a fat function that describes the likelihood of any point to belong to a fat layer. The fat function is diffused from the skin surface using the fat scalar value defined by the user. And this function goes to 0 at any of the points that belong to any of the muscle curves. So to compute all these muscle functions and the fat function, we propose to define them as the minimizers of the Dirichlet energy subject to constraints. So we define M as the total number of muscle curves, and we sum the Dirichlet energies of all the muscle functions over our domain. Here, this matrix A of I is an optional user-defined diffusion tensor field, which we could use to bias the direction in which the material flows at any point in space. And since the fat diffuses isotropically, we just write it as a separate term. And here are constraints. So the first constraint ensures that we respect the user-specified tissue values at the muscle curves. And the second and the third ensures that each muscle function goes to 0 at the skin boundary and at the points that belong to other muscle curves. And finally, the last 2 constraints ensure we respect the user-specified fat values at the skin boundary and that it goes to 0 at all the muscle curves. So we showed this an example in 2D, but basically in 3D, we were working with the tetrahedral meshes, but the formulation of our optimization remains the same. And in practice, the way we generate this TET mesh is we just take the triangular meshes of the skin and the bones and just run them through TETGen. So now assume that we diffused all of our tissues and we end up with some scalar value per every point in our volume. However, each point in our volume can only belong to a single tissue. So the points that belong to the bone tissue are simply all the points that are contained inside the tetrahedrons comprising the bone geometry. So we don't need to do anything there. We get that for free. We only really need to find a way to differentiate between different muscles and fat. And the way we do that is actually quite simple. We treat the muscle and fat functions as probabilities and assign the point in our volume to the tissue with the highest probability. So now we know which tissue does any point in our volume belongs to. So now that we have this segmentation, how do we visualize this in 3D? In the end, we only want to visualize all points belonging to the muscles, while the points belonging to the fat should just be invisible. One thing we do not want is to perform any remeshing operations when the user creates or edits the muscle curves, because that would be just too expensive to compute and will basically affect the interactivity of our tool. So another way to handle this is with volume rendering using fragment shaders. I won't go into more detail, but here the line image that you see on your screen is an example of a volume rendered muscles. So these are not shapes that are like we extracted and then use some kind of polygonal rendering to visualize them. This is all volume rendered. So given the complete muscle curve network and the corresponding muscle functions defined over the volume domain, we now need to extract the geometry of the muscles so that it is suitable for downstream applications. So isosurface extraction methods do not trivially solve our problem because we're not simply extracting an isosurface of one scalar function. Instead, we want to compute a pointwise maximum of multiple scalar functions over our volume, and we want to extract the boundary that separates each function. And we cast this problem of extracting the muscle surface geometry as a solution to the computation of the upper envelope of our tissue functions. So first, let's look at a simple example of an upper envelope problem in 2D. So we have here 4 line segments that are defined over our 1D domain from A to B. And the upper envelope is the pointwise maximum of all segments over the domain. Here it's highlighted as a dashed blue line. And the maximization diagram is the subdivision of the domain into multiple cells, where each cell's identity is induced by the upper envelope. So alternatively, we can think of the maximization diagram as the projection of the upper envelope onto our domain. So let us now define the general upper envelope problem. So let a set S be n d simplices in d+1 dimensional space, where a d simplex has d+1 vertices. So in other words, d of equal 1 is just a line segment, d of 2 is a triangle, and d of equal 3 is a tetrahedron. We can those view each S of I as the graph of a partially defined linear function. Then the upper envelope of the given simplices is the pointwise maximum of all of these functions. And the maximization diagram is the subdivision of our domain into connected cells obtained by projection of the upper envelope onto this domain. And if you remember how we formulated our volumetric segmentation problem, then you will see that it basically is identical to solving the upper envelope problem over our volume. And in our case, we're interested in finding maximization diagrams of all tissue functions over the volume constrained by the surface of the skin. This problem can be solved by considering finding the maximization diagrams of the tissue functions over each tetrahedron separately. So here on the left, you see an example of the maximization diagram of the functions defined by the scalar values at the vertices of the tetrahedron. And on the right, you can see basically us tetrahedralizing those cells. So as a result, the original TET is split into more subtets, where each subtet has only one tissue function that is the maximum at that TET. Here's just a simple example of us taking a uniform 3x3 tetrahedral mesh cage with 2 scalar functions defined over it and just splitting it into 2 tetrahedral meshes. So the split is the result of computing tetrahedralized maximization diagrams over every tetrahedron. So now we are ready to define an algorithm for computing tetrahedral maximization diagram. Due to time constraints, I'll just simply go over kind of a pseudocode just to give you an idea of what it looks like. So our input is a tet mesh defined by two matrices. The T is the indices of our tetrahedrons, V are our vertices, and we have a bunch of scalar functions that define over our mesh. And our output is just simply another tet mesh where we have this additional matrix L that tells us the identity or the label of each tetrahedron. And labels basically tell us which tissue does this tetrahedron belong to. And so this is the pseudocode, but the main thing I wanted to point out is this split tetrahedron function, which takes a single tet and two different tissue functions and just computes the maximization diagram of those two functions over the tet. And kind of the main point of the slide is just to show you that the implementation of the algorithm is not complex. It's just like a bunch of for loops, and split tetrahedron under the hood just requires a bunch of edge splits. So it's not difficult to implement. So here's just an example of us running this algorithm over the line example. And you can see that because our volumetric domain is represented as unstructured mesh, we could potentially get not a very nice topology. However, we do guarantee that the tetrahedral meshes are manifold, so remeshing them is not an issue. And here are just some of the rendered results that I wanted to show. Hopefully you can also see fiber fields overlaid. And I don't think I have time for limitations, so I'll just say thank you very much for listening. Yeah, that's it for me.
Thanks, Rinat, for that lovely presentation and for all those lovely illustrations. As for questions, if you're a longtime fan, we usually keep the questions until the end and have a joint Q&A session. If you have any further questions after that, we do have a Discord server that we encourage you to join and post to, and we will guide the authors towards that Discord server as well. Now, I'm very excited for our next talk as well. The reason being, many of us organizing this colloquium or watching this colloquium in our field generally understand the fact that geometry is everywhere. It's all around us. Maybe we were told this in high school, or maybe we just know that from our experience, but still, it's still very easy to confine our idea of geometry processing and the use of studying geometry to a very narrow topic. It can be very easy to stay focused on the applications you're working on. And our next speaker's line of work really does remind me of the absolute breadth and applicability of our field. Like, how many times have I actually thought of the geometry of a human organ, like really? And how does the geometry or shape of a human organ actually affect medical conditions or medical mental conditions? What about at a smaller scale, like proteins or protein structures? And before even getting into this cause and effect problem, how do we even begin by describing the geometry of such complex structures? She's a professor at the University of California, Santa Barbara, where she leads the BioShape lab. And I'm very excited to learn more about such an interesting intersection between geometry and biology. Please welcome Professor Nina Miolane.
Thank you very much for this very, very nice introduction. I'm also very excited to be presenting my research today. So I'm going to talk about exploring the geometries of life, which means that we want to be able to look at all the beautiful shapes that different biological structures adopt in our bodies and be able to say how these shapes are actually related to the functioning of the organisms and how they can reveal new insights on different pathologies. But before, let me state the overarching goal of this research, which is a type of biomedical research. And our goal there really is to understand the mechanisms of life. And today we are very lucky to understand the mechanisms of life because we have a unique opportunity, which is that we can just look at them. And by that, I mean that we have access to different types of bioimaging modalities at different scales. So from the macroscopic scales, which is the scale of organs, but all the way down to the nanoscopic scales, which are the scales of proteins. So let me give you some examples of bioimaging data that we can work with to understand the mechanisms of life. So first, at the macroscopic scale, one very interesting imaging modality is called MRI. It stands for magnetic resonance imaging. It's actually an imaging modality that got the Nobel Prize in 2003. And it's a remarkable imaging modality because it allows us to image a type of biological structures, so organs in that case. But actually, all the way to the nanoscopic scale, the advances of bioimaging techniques have allowed us to look at smaller and smaller biological structures. And more recently, at the nanoscopic scale, we have cryo-electron microscopy, which is an imaging modality that allows us to look at proteins or different biomolecules at a near atomic scale. And for this reason, this imaging modality received the Nobel Prize in 2017. So to understand the mechanisms of life, we have an incredible source of data, which is these bioimages. And the point I want to make in this presentation is that within all of these bioimages that we have available from the macroscopic scale to the nanoscopic scale, there is a type of information that's very relevant to understand the mechanisms of life. And this information is the shape information. So let me give you examples. At the macroscopic scale, first, let's look at organs. On the screen, you can see 3 slices of 3D brain, which is undergoing atrophy. You can see that the gray and the white matter are losing volume as the years go by. And conversely, the ventricles, which are the black ovals in the middle of each brain slice, they are gaining volume. And the reason for that is that the brain that you actually see on the screen is the brain of a subject with Alzheimer's disease. And Alzheimer's disease is an accelerated aging that causes this atrophy of the gray and the white matter. And so we see already that at the macroscopic scale, by observing shapes of organs in images, we have a correlation with the state of a patient, which in that case is the state of a disease, Alzheimer's disease. So that's for the macroscopic scale. But actually, all the way to the nanoscopic scale, and even if we stay with Alzheimer's disease is actually a shape problem, even at the nanoscopic scale. So on the bottom there, you can see the beta amyloid structure and the tau structure. And it has been shown recently that Alzheimer's disease is actually a double prion disease, which means that this biomolecular structure actually change shapes. And by changing shapes, they have an influence on the cellular biology of organisms. And more specifically, as you can see in this video, you see that the tau, they aggregate near the neurons. So they form these blue aggregates, and the beta amyloid also aggregates. And so as a consequence, they block the normal functioning of our cells, which in turn causes Alzheimer's disease at the macroscopic scale that we have on the top of the screen. So from the macroscopic scale to the nanoscopic scale, from the bioimages, there is one semantic information of importance, and this is the shape information. So I hope it's clearer from the previous slides that both the function of a biological structure, but also its state as healthy versus pathologic influences the geometry of that structure, should it be organs, cells, or proteins. But what about going in the reversed way? If we have access to the geometry thanks to these bioimaging modalities, can we use this shape information to make new biomedical discoveries? Can we advance fundamental research in biology and medicine up to the point maybe where we can give automatic diagnosis of the disease of interest? So at the nanoscopic scale, but also at the mesoscopic scale with the shapes. So this is why I'm interested in doing shape analysis from biomedical imaging, not only looking at the pixel level and intensity information, but really extracting this notion of shape from the images. And when we do research projects in my lab, usually we start with a biomedical question. Then it means that we need to acquire the images. So that is done by the collaborators we work with. The next step is to extract the shape from the image. So performing shape reconstruction, which is sometimes not a trivial process, because sometimes the 3D shapes comes in different 2D in a set of different 2D images that are very, very blurry, and just obtaining a shape information in 3D is a challenge. Then there is the question of how we model the shape in a computer from a mathematical perspective, a computational perspective too. How we do statistics to be able to finally answer our biomedical question. And I want to insist for a second on how important it is to have a good statistical theory of shapes for these applications. And this is precisely because we're working in the biomedical fields where we don't want to make errors, because downstream there are important consequences on biomedical knowledge and sometimes about the lives of the subjects and patients at hand. So having a strong statistical theory of shapes is very important there to be able to quantify the certainty associated to each type of finding. So this is the overall flow of how we can use shape analysis to answer a biomedical question, starting from biomedical imaging data. For this talk, I'm going to focus on two steps of this flow, which are namely shape modeling and shape statistics. So let's start with shape modeling. So on the screen, you have a beautiful image of a cell, which is moving. And the question there is how, beside the fact that it's inherently beautiful, how can we quantify what is happening there? What is the mathematical definition of a shape? How can we represent that in a computer? And then how can we come up with statistical models that can describe the variability in shapes, but also the uncertainty we make when we compute with shapes? So let me give you a fast introduction on the mathematics of shapes and shape deformation or transformations. So first on the left, you see a cell shape. It's actually a shape of a cell with bone cancer that is undergoing 3 types of transformation: scaling, translation, and rotation. Now, it always depends on the question that you want to answer. But let's say for this presentation that these transformations are the ones that do not change the shape. So when we translate a shape, that doesn't change the shape. When we rotate the shape, it doesn't really change the shape. And then the scaling, depending on the application, can be considered shape or not shape. Here in this case, we're going to consider that rescaling a shape doesn't change the shape itself, that the shape is not tied to a notion of size. So with this formulation, basically what we're saying is that a shape, from a mathematical perspective, it's everything that remains invariant about this object when we have removed the effect of the translation, the rotation, and the scale. So that's a shape, that's an equivalence class, putting it in mathematical terms. So to introduce a few mathematical notations, basically what we're saying is that there is a group, G0, a group of transformation, the group of translations, rotations, and scaling, for example. More precisely, it's what's called the Lie group. It's a group because the identity transformation belongs to the group. You can compose 2 transformations and you still get a transformation, and you can get the inverse of a transformation. So it has all the good properties of a group. It's also a Lie group because it's a continuous group there with some smooth differential structure. So in order to represent shapes from a mathematical perspective, we define this group, G0. Then next we define the action. It's called the action of each element of this group. So small G0 on X, where X is the original object, whose shape we want to describe. And then we said that the shapes themselves are the equivalent classes under the action of that group G0 on the original objects. And so if you want to look at how we describe that from a mathematical perspective, we say that the shapes are an element of a quotient space, Q, where Q is defined as the set of the equivalent classes. So equivalent class of X is denoted with this bracket. And you have the mathematical definition of the equivalence class of X, which is what I said before. The equivalence class of X are all the other objects, Y, that have the same shape as X. And what does it mean to have the same shape as X? Well, it means that there exists an element G0 from the group of transformation G0 that doesn't change the shape, such that we can transform X into Y. There is one translation that relates X to Y, or there is one rotation that relates X to Y, etc. So that's how we go from an intuition about what is a shape to a mathematical model using this concept of quotient space. So now when we want to do statistics or learning on shapes, we need to do statistics or learning on these equivalent classes, which are elements of a quotient space. That's for a first model of shape. Second model of shape is using kind of a differential perspective and saying that we can describe the shape as a deformation from another shape. And so in that case, the group that we use, so we define a new group G, that's going to be the group of smooth deformation. Again, it's a group because the identity belongs to it. You can compose deformations. You can take the inverse of a deformation, and it's a continuous group. It's a Lie group. Now, the elements of these groups are precisely the ones that describe the shape. And so we don't need to do all the quotienting that we were doing before. For this one, the group itself represents all the possible shapes. And so in this case, the shapes are represented by the deformations, which are the elements of the Lie group itself. So we will use small g belonging to this new group to represent the shape. So these are the two main models of shapes illustrated here on cancer cells. So what do they have in common? There is something very, very interesting, one that happens once we want to do statistics on these shapes, is that whether we use the quotient space approach, meaning we remove everything that does not change the shape, or we use the differential approach, meaning we see a shape as a deformation from a reference shape. In both cases, we end up with shape spaces, data spaces, that have some interesting geometry. And specifically, we call them manifolds with additional geometric structure. So for our talk in the geometry colloquium, it's very interesting because the geometry appears at two levels here. First, we have the geometry of the shapes themselves, but now the data space that we end with, which is a quotient space or a Lie group, also has an interesting geometry. And the fact that it has such an interesting geometry means that every type of statistics or learning that we want to do on it needs to be rethought a little bit. So let me talk a little bit about manifolds, which are these type of data spaces that can represent shape spaces. So quotient and Lie groups are manifolds. What that means is that if we want to compute on quotient and Lie groups, which means if you want to compute on shapes, we're going to have to compute on manifolds. And I'd like to insist that we need to generalize computing, statistics, and deep learning to data on manifolds now. I'm going to give an example of why you actually need a new theory to do all of the usual learning that we know how to do on traditional data, to be able to do it on manifolds, therefore on quotients and Lie groups, and therefore on shape data. So let me explain a little bit what is a manifold. So we can explain it roughly by giving the difference between a manifold and a vector space. So on the left of the screen, you have example of data on a vector space. You have these 4 blue points that live on a vector space of dimension 2, which is the plane. And the definition of manifold that we consider here is a manifold is is a space that is basically a vector space that is allowed to be curved. So locally on the manifold, if you go very close to each data point, the space looks flat. But if you take a global view, then the space is allowed to have some curvature. So again, now I'm talking about the geometry of the space of the data. And so this is what is illustrated in the center of the screen, where you have these 5 points that live on this curved surface, which is an example of manifold. The easier example of manifold that we are used to is the manifold that describes the Earth. In other words, a sphere. And so a very simple example of data on manifold is any type of geographic data. For example, the coordinates of cities on the Earth, as you have illustrated on the right, coordinates of earthquakes, etc. So beyond the example of shapes that belong to quotient spaces or Lie groups that are examples of manifolds, you also have other data that belong to manifolds. So for the next slides, I'm gonna just go away from the shape cases just to introduce the geometry of the data space. I'll use the sphere as the example, and we'll go back to the shapes just after. So in order to generalize computing, statistics, and deep learning to data on manifolds, I'm co-developing an open source Python package that's called GeomStats, which stands for Geometric Statistics. So why do we need to generalize computing, statistics, and learning on manifold? Let me give a simple example, the example of the mean. Which is arguably the first step of any statistical analysis, computing the mean of the data, because this is the point that summarizes the data set. So if we look at the definition of the mean, x bar, we see that it's a linear definition in the sense that it's a weighted sum of the xi, which are the elements, the data points. Now, from the previous slides, we know something about the geometry of the data space. The geometry of that manifold is that it is allowed to be nonlinear. So there is a conflict there because we're trying to use a linear definition on a space that is nonlinear. And the consequence of that conflict is that if you use a linear definition of mean, the traditional definition of mean, you obtain a mean that may not belong to the manifold. So as I said, I'm exemplifying this on the sphere because it's more visual, but we'll go to the shapes after. So what I mean is that if I have 2 points that live on this manifold, which is represented by a sphere, and I use the traditional definition of mean, I end up with the orange point that does not belong to the manifold. And so from a shape perspective, it means if I take the usual definition of mean on 2 transformations, I'm going to get a mean that is not a transformation itself. So if you take the linear mean of 2 rotations, you're not going to have a rotation in the general case. And that stands for any type of statistics that we want to do on the shapes. On the left of the illustration, you see a code snippet that's from the GeomStats package that allows to generalize these statistics. And so just parsing this code a little bit, before getting into other types of code, you can see that the GeomStat package has a geometry module that allows us to import a hypersphere manifold. And the sphere is just a hypersphere of dimension 2. And so this is the code snippet that allows to produce the figure on the right. All right, so the traditional definition of mean does not work for shape data because they belong to manifolds and these manifolds are curved. And so the way we can generalize this definition 2 points on manifolds is basically taking a property of the mean on vector spaces and making the property of the mean the new definition for our manifold. And so here we define the Fréchet mean that has been defined in the 1940s by saying that the Fréchet mean of points on the manifold is going to be the point that minimizes the sum of the squared distances to the data. So we need a notion of distance on a manifold, which comes from a notion of metric. And when we have this, we can say that the mean is going to be the point on the manifold, so it's constrained to be on the manifold, that minimizes the sum of the square distances to the data. So without going into the mathematical details, I'm showing you on this code snippet how it can be implemented with GeomStats. So before I was introducing the fact that geomstats had a geometry module that was allowing us to get different geometries, meaning different types of manifolds that represent data spaces. Here you see that it has a learning module in which there is a Fréchet mean, which is the generalization of the traditional linear definition of mean. And without going into the greedy mathematical details, we see that we can use the Fréchet mean estimator on any type of geometry represented by a metric, which gives the notion of distance. And we can fit this estimator to the points to get an estimate, which is now this orange point on the right, which represents the Fréchet mean of the two blue points. And now it belongs to the manifold. So at that point, I'd like to make two remarks. First is that you may think that this is too complicated in the fact that the sphere, for example, can be embedded in the 3D space. So why not just computing the mean in the 3D space and then projecting that back to the manifold if we need to? Why not doing all the statistics in the embedding space that embeds the manifold and then just implementing a projection? And this is a valid remark because there is a Whitney embedding theorem that says that any manifold, these curved spaces, can be embedded in a vector space of dimension 2m, so up to 2 times the dimension of the original manifold. And actually in this dimension information, there is the first hint of why we didn't want to do all the statistics in the embedding space and then project back. And this is because in the general case, we double the dimension of our data. And this might lead to a numerical slowdown. And so it seems from a mathematical perspective, it's kind of a loss. It seems that it's unnecessary increasing the dimension of the data. And the second argument is also that, for example, with the case of the sphere, of course, we can compute in the 3D space, but it means that we need to operate with X, Y, Z coordinates, which are not very intuitive to represent elements on the sphere. We'd rather work with longitude and latitude that are more interpretable. So these two arguments corroborate the idea that we want to compute directly on the manifolds without having to think of it as embedded in a possibly high dimensional space. And the second remark that I want to make is that here I illustrate everything on a sphere to represent, to explain what are statistics on manifolds, which are these curved spaces. But in the case of shape spaces, the manifolds that are the quotient spaces or the Lie groups, they are curved, but they also have additional properties. So they have a group composition, they have some types of symmetries. So not only we need to generalize geometric statistics to geometric statistics to curved spaces, but we also need to respect the additional geometric properties that these specific spaces have. So I'm not going to talk about the group composition and elements like this, but keep in mind that this generalization of linear statistics to statistics on curved spaces also extend to statistics on curved spaces that have some specific geometric properties. So the output of this research is this software called GeoMstats. Again, it's to compute, do statistics, and possibly deep learning for data on manifolds. With the idea that deep learning is possible, it comes with 4 different backends, including some that are tailored to the deep learning applications. But these backends are also very interesting because 3 of them have automatic differentiations. And deep down in the core of this software are elements of differential geometry. And because it's called differential geometry, we can understand that it's using differential operators. And so it's very interesting to have these backends to be able to write down all the equations of differential geometry easily. And if you want to use GeoMstats, is as we saw before, you apply 2 steps. First, you define the manifold of interest. In our case, it was a hypersphere. And then you apply the estimation or learning method that you want to use for your data. In our case, it was the Fréchet mean. And GeoMstat was created with 3 main objectives. The first one is to teach hands-on geometric statistics and learning. So even though there are a lot of very good textbooks on differential geometry, there are a bit less on differential geometry that couples geometry statistics and that come with hands-on exercises. So we'd like GeoMstats to be a companion, a code companion for all of these textbooks. The second objective of GeomStacks there is to democratize the use of geometric statistics and learning. So I completely hid all the computation of differential geometry into these two structures that you can see here, the hypersphere and the Fréchet-Min. So from a non-expert, non-differential geometry expert perspective, in these 3 lines of code, all the baggage of differential geometry is readily available. And we use the API that is a scikit-learn API, meaning that it should be easy for a machine learner or someone from computer vision to use this method out of the box because it's the same API as scikit-learn. And the third objective is to support research in geometric statistics and learning, because there is a growing community of applied mathematicians, machine learners too, statisticians, that contribute this method that allow to generalize not only the Fréchet-Min, but all different types of statistical learning algorithm to manifolds and possibly manifolds with additional structure. And personally, I use it to compute with shape data. So let me give you an example of what you can, 2 examples actually, of how you can use this dichotomy of first defining the geometry and then defining what you want to do with the geometry to compute on manifolds, and specifically the manifolds that are linked to the shape data. So when we describe shape data, we say first, oh, we can transform shapes with transformation that do not change the shape itself. So on the left here at the beginning of this code snippet, you see that I import one geometry, which is called a special Euclidean geometry. And the special Euclidean geometry is actually a Lie group. It's the Lie group of translations and rotations. So here I'm working with this first set of transformations that do not change the shape. So let's say, for example, I want to compute a geodesic, which is the path between 2 of these transformations, so on the Lie group of rotation and translations. I instantiate the object SE3 as the special Euclidean group of translation and rotation in 3 dimensions. I extract its geometry, that's represented by these metric objects that allows us to compute distances and also curves between 2 points on the Lie group. And then using this metric object, I can compute what's called a geodesic. So on my curve manifold, which here is the Lie group SE3, I compute the curve that allows to link 2 points on that manifold with the shortest path. So this is a geodesic, and this is what you have illustrated on the right. So you see pose 1 is a little frame, which has 3 arrows, a blue, a green, and an orange. A frame, it's an origin and an orientation. So it's a translation and a rotation. So a frame placed in Euclidean space represents a translation and a rotation, so an element of SE3. And you can see on the screen a geodesic that links 2 elements of SE3. And then we can look at deformation of shapes, in that case, cells. So what happens when I have removed all the rotations, translations, and scaling that can affect the object, and I look at what remains, which are the equivalent classes. And so you have the code that allows you to do this on the bottom left. So the geometry I'm interested in is the geometry of discrete curves. I instantiate curves, which is an object of the class discrete curves in R2. So the cell comes from 2D images, so 2D plane. I instantiate the data space of curves. I extract the metric that I'm working with. And this is a metric that allows to compute similarities between cell shapes when we have removed the action of translation, rotations, and scaling. And then I compute a geodesic according to this metric, and this is the geodesic that you can see on the right there. And this is a geodesic in the space of shapes now. So for the sake of illustration, the 2 shapes are aligned so that we see that it's a geodesic in the shape space of shapes, but this geodesic does not depend on rotations, translations, and scaling information. So I exemplified first this kind of technique on the sphere because it's easy to visualize. It's a 2D manifold. We see the curvature, etc. And then I gave two other examples on the data spaces that are manifolds of Lie groups and quotient spaces that appear when we deal with shape data. So not only these spaces are curved, they're also higher dimensional, not only dimension 2, and they have additional geometric structures. But beyond the original very simple example and these two spaces, actually there are many manifolds that represent different type of data that arise in the biomedical imaging field. And in the GeomStat, we have implemented all the geometries in the sense of the geometries of the data spaces that arise there. And we use an object-oriented programming in the sense that you have a parent class that's called manifold, from which all the other abstract classes inherit. And then you can go, you go down the hierarchy until you reach an actual manifold. So for example, here on the right, you have spatial orthogonal, just the rotations of any dimension, spatial Euclidean, rotation and translation of any dimensions, etc., etc. So that's from the geometry perspective. But of course, there is the learning perspective too. So I mainly talked about how we generalize the Fréchet mean, the mean to the Fréchet mean. But actually, if we need to generalize the mean, which is the very first step of any statistical analysis, we can understand that not only the mean needs to be generalized, but also all the different algorithms that we have in statistics, learning, but also probability theory. So on this table, you can see an overview of my research, which is basically aiming to fill all of these gaps and asking the questions, what happens when I change the geometry of the data space and I try to use the traditional statistical learning technique. So in terms of geometry, the type of geometries that we saw are Lie groups and quotient spaces, because these are the ones that arise when we deal with shape data. But there are other types of geometry that we can consider. Riemannian, Finster, affine, stratified spaces also appear when we deal with shape, sub-Riemannian geometry, etc. And the columns there represent the learning algorithm, so or statistics procedure or probability theory elements, point estimation, dimension reduction, stochastic processes, etc., etc. So you can imagine that we want to generalize all this algorithm to the different types of geometry and also understand if the theoretical result that we had in the linear case still hold for these cases. Do we have a central limit theorem? Do we have results like this? I'm going to give you an example using shapes on how we can use all of this to actually answer a biomedical question. And I chose this one because this is the most recent work, and also it uses the structure of quotient space that we've seen to describe data spaces of shapes. And it's fairly easy from a learning perspective because we're going to do point estimation. We're just going to estimate the mean. So this is the traditional flow. I talked about how we can model shapes. I talked about the need of generalizing traditional statistics to shape statistics. Now let me show how we can use all of this and actually simple elements from shape modeling and shape statistics that we have seen to answer a biomedical question. All right, so the goal is to understand the impact of drug treatment on cancer cell shapes. So on the screen, you have images of 3 cells that are bone cancer, actually. And so from the recent literature in biology and tumor grading, the grading of the tumor of the cancer is still mostly done by human experts. And what this paper was showing is that this can have low accuracy and maybe also more worryingly, a problem of reproducibility, meaning that 2 different human experts trying to grade the cancer, meaning trying to estimate how serious the cancer is, will give 2 different answers. And so where automatic shape analysis is interesting in this setting is that we can democratize this process, Because if we manage to have an algorithm that can extract biologically and medically relevant information from the images themselves, then we're going to create an assistant, a computational assistant to the human grader that will be consistent. And so we will have something that's reproducible because each time this algorithm will give the same result. And not only this, if the algorithm ends up having good accuracy, then we can share it because we can theoretically copy paste the software and share it everywhere. So it allows to democratize biology and medicine in that respect. So for this application, we look at cell morphometrics. So the state of the cell in this case is the cytoskeleton of the cell impacted by the drug. So if it is impacted, then the cell shapes should vary. And this is grounded in biological knowledge because when we give drugs to this cell, it modifies the actin activity, which changes the perimeter of the cell. So we know that there is a link from the cell state to the cell shape. Now we'd like to use the tool from geometric statistics to do the reverse, to say, if I see a shape, can I infer the state of the cell? And this raises the questions, how can we quantify differences in cell shapes? And also, how do these different cancer treatments affect the cell? In the sense, how do they affect the shape? And how can we deduce, what can we deduce on how they affect the state itself? So this is a data set of cancer cell shapes that we have available to tackle that question. By the way, in GeomStat, so the software that gathers research in geometric statistics, we make some data sets available to the community. So a little bit in the same way that you have IRIS data set in scikit-learn, we provide some example of data sets on manifolds so that you can have an idea of what type of data belongs to manifold beyond the shape data. And so this data set, actually, we make it available on GeomStat. So you use the datasets module of GeomStats, and you can just load cells and you'll have access to this data set. So this data set is a data set of 2D curves. Some are controls, and then the two others have been treated with two different drugs. So I won't go into the details, but the first drug is called CYTD, and the second one is called JASP. And we'll like to be able to quantify what we are starting to see qualitatively on the screen, which is that it seems that the CYTD treatment has an influence on the cell shapes, because you see that they become more and more irregular compared to the control. And then the JASP treatment also has an influence, but not that much for the blue cells. The blue cells come from a different type, different stage of cancer, but it does have an influence on the orange cell, which come from the dun line, which is the cancer is at a different stage. So that's something that we can see visually, but visually might be not enough because this is only a subset of the data set. And also, again, we're in the biomedical field, so would like to be able to quantify what we're seeing and also give some notion of uncertainty on the results. So this is how you use GeomStats to answer the question. Our idea is that we can just compute the mean shape and then quantify how different are each of the cells from the data sets from the mean shape. And by how different, I mean a notion of similarity that only concerns shapes and is not confounded by translation, rotation, and operations like this. So we use GeomStats and actually both the geometry and the learning that we've seen already. So I instantiate a curve manifold as a manifold of discrete curves on R2. You can instantiate for other application, you can have discrete curves on the sphere or another type of manifold. I define the learning algorithm that I want to use, which is the Fréchet mean. So this generalized definition of mean. Which geometry do I want to use? I want to use the geometry of the curves using the square root velocity metric, which is a notion of similarity that only focuses on shape. and I can compute the mean estimate. So in this case, in the case on our data set, this is what you get. So in black, you have the mean cell. And when you superimpose that on all the points that define the discrete cell, so the boundary of the cells discretized into a finite set of points, you see that we get a shape that makes sense. And now we can answer the question using hypothesis testing on the magnitude of shape transformations. Or the shape transformation is how each cell of the data set was the magnitude of how each cell of the data set of the transformation that transform each cell of the data set to the mean shape. So these are the histograms that we get. So for the control cell, we see that there is in the blue cells, they are not that far from the mean shape, but on the orange class, they have more variability first, but also further away from the mean shape. And now it's interesting to see what happens when we treat these cells. So if we treat the blue cells with JASP, we see that the distribution doesn't change much. Whereas when we treat the orange cell with the drug JASP, then this orange cell distribution is shifted away from the mean shape, but also is mostly shifted away from the mean cell. And then when we treat with the second drug, with CYTD, we see that now this drug has an effect on those two stages of the cancer. So the stage represented by the blue cells and the stage represented by the orange cell. So using this simple similarity metric between cells, we can see that in the second stage of the cancer, which is from the dun line, the orange cell, there is a higher variability in shapes. They are further away from the mean shape. We see that the JASP treatment is very effective for the cells that are in dun stage, less effective on the DLM8 cell line, so the other cancer stage. And the CYTD treatment is very effective on both. And here I'm showing the histogram, but you can back that with hypothesis testing and actually give a notion of statistical significance for this result, which is what's needed for the biomedical applications. So in summary, I'd like to show you again the traditional flow of shape analysis for biomedical imaging. We start with a biomedical question. We need to acquire images. This is done by our collaborators mostly. The shape reconstruction stage, I haven't talked too much about it, but it's sometimes a very challenging step, especially when you're dealing with images at the nanoscopic scale that are very noisy. It's very hard to just see the biomolecules there. And then today I've talked about shape modeling and shape statistics mostly, and specifically in the example on how we can link the geometry of cell shapes to the impact of drug treatments, cancer drug treatments. And from a methodological perspective, what I'm interested in is generating statistics and learning on data spaces that have different geometries. And even though I focus on the case of shapes, it's interesting to note that this property of Lie groups and quotient space that arise in shape statistics and shape learning actually bear more fundamental concepts that are the concept of equivariance and invariance. And these are the concepts that are used today to define the field of what's called geometric deep learning, which is a geometric framework to explain why convolutional neural networks or other deep learning architecture work and do not work. It's explained by geometric properties on these specific spaces. And with that, I'd like to thank you very much for your attention, and I'm open to questions.
Well, thank you very much for that lovely presentation, Professor Miolane. Right now, we're going to enter our Q&A session right now. We can start with a few questions for Rinat so that we can give Professor Miolane a break. Maybe get some water or something. Rinat, we have a lot of questions for both of you, I suppose. Are there any requirements on the connectivity of the bones in your algorithm? Are the just anchor points for the muscles? And what exactly is the role of the bones apart from that?
Right. So yeah, the bones, the connectivity doesn't matter, and they actually just serve more as like a boundary constraint so that the muscles don't overlap, or like they're not being sculpted in places where the bones are supposed to be. So yeah, they do serve basically as an anchor.
Oh,
In fact, during the post-processing, when we do the TET meshing, we actually, after we TET mesh the skin and the bones, we actually remove the bones from the TET mesh. So all the diffusion happens on the mesh without the bones. So they basically serve as like another boundary constraint for our diffusion. Yeah.
You see, for your volumetric diffusion. Very cool. Let's see. I'm wondering, have you ever thought about how well this process approximates the geometry of real world muscles? Are there times where you can note this muscle shouldn't have this shape, or this muscle just looks wrong?
Yeah, that's a great question. So, because I didn't have time for my limitations section. So in that slide, we actually talk about that one of the main limitations is that if you open up an anatomy book and you look at the muscle shapes, they very often don't just have like two attachment points, right? Or like one point on each end. They actually have many attachment points. For example, like our chest muscles, and so modeling those right now is not possible because we do represent a muscle with just a single curve. So our muscle system is more for like a fusiform shaped muscles.
Or what? Sorry, could you repeat that?
Fusiform. Fusiform basically is like what you see in my modeling too. That's like the main shape that we handle. I guess because our primary application of this tool is for muscles for like visual effects and gaming. So we can kind of get away with not creating like a very anatomically correct muscles. But we do think that if you just simply represent a single muscle with multiple curves, you can actually work around this limitation. We just haven't tried yet. So it's a future work.
Thank you very much. We have a few questions for Professor Nina Miolane. So let me just find them. The first thing I'd like to do is re-clarify something that you said that was that led to a really interesting idea. That being that you're learning, the deformations that you're learning both have a geometry, but they also live in this space that itself has a geometry. Could you talk a little bit more? Could you talk a little bit more about this space that they live in, and specifically what type of space do regular cell deformations live in usually? Just recap that entire idea, because it sounds very, it really messed with my head for a little bit.
Yeah, and thank you for this question. This is actually an important point. Because when I talk about geometric statistics, actually this geometric word has a double meaning that, as you said, the data points are shapes. And so they have the geometry of shapes, which is the traditional sense that we use to talk about geometry. But as a data point on a space, which is a data space now, the data space itself has some geometry. So about the geometry of the data space, which is the space of the data points, which is the space of these shapes, what is the geometry of that shape, of that space? So in the case where we represent shapes with deformations, the space is a Lie group. So one shape is represented as a deformation. One deformation is a point on the Lie group of deformations. And so what is a Lie group? It has some geometry, which means it's curved, but it also has additional properties in the sense that you can imagine 2 points on, you can imagine 2 points on this Lie group, so 2 deformations. You can compose them and you get another point. And there is 1 point on this data space that is the identity. So that's very special. There is one very special point on this data space, which is the point that corresponds to the identity deformation, meaning the deformation that doesn't deform anything. And so you have more symmetry on this data space. And if you want to describe this data space with geometric elements that respect these symmetries, then it's going to give a shape to the data space itself. So for example, in the case of the Lie group of 3D rotations, if you define a way of measuring distances between points on this data space, which are between two rotations, if you're on a distance that respects the symmetries of the Lie group, it's going to give a shape to the data space itself, which is going to be more or less a sphere. It's a double cover of a sphere. And so it's very interesting to see about the dual meaning of the word geometry there, especially for this colloquium, which is both the geometry of the shape that is now one data point that belongs to a data space that now has geometry and shape itself.
Yeah, definitely. This sounds very useful for any sort of prediction of deformations on shapes in general, which is a very popular field right now, in graphics as well.
Mm-hmm.
Let's see what a good follow-up question for this is. Is there any work on determining what type of spaces cells deform within? So I guess you talk about how a deformation can lie in this deformation space. What parts, is there any work on determining what sort of spaces these cells deform into?
Yeah, so again, very, very good question. So from a mathematical perspective, all the deformations are possible in the sense that we model one deformation as an element of the Lie group of diffeomorphisms. So I haven't used that term in the presentation, but let's call them diffeomorphisms. So one diffeomorphism is a smooth transformation. And the Lie group of diffeomorphisms from a mathematical perspective is that it's an infinite dimensional Lie group. So we'll need to put that in the computer, and there are some specific computational questions there, but assume we can have a finite dimensional representation of this infinite dimensional Lie group. Now, from a mathematical perspective, one shape is going to be one diffeomorphism, is going to be one element of that group. But the question that we want to ask is asking, okay, these are all the possible mathematical deformations. What are the ones that can shape use? So in other words, what is the subspace? So in this large Lie group of all possible deformations, can we learn the subspace of the deformations that are adopted by cells? And so that's a dimension reduction question from a statistical learning perspective, the PCA, basically. Except that, as we saw, we cannot just use PCA because the space is curved. And so we need to generalize PCA to the Lie group. And I think the generalization of PCA is an interesting question because in a linear space, PCA is PCA. On a manifold, there are at least 4 or 5 ways that you can generalize PCA, and they all make sense and they all boil down to traditional PCA when you're on a vector space. So you pick one of the PCA, generalized PCA technique, and then you can see which type of deformations can be adopted by shapes. And I think one of the most interesting questions there is then, can you link the principal axis of deformation to biophysical variables? And very often you can see that it's the case. So traditionally, taking the example of cells, but also maybe organs, it's easier to see the first axis. If you haven't quotiented the size, the first axis of the generalized PCA is going to be the growth, like the BMI, body mass index, the size of the person. And then other axes usually are going to correspond to different medically interpretable or biophysical variable. So it's very interesting to see that you can recover the medicine side or the biological side by a dimension reduction technique that was tailored to the type of data space that we are using.
Mm-hmm. Absolutely. Maybe one more question for Professor Nina Miolane, and then So we'll go to Rinat with a few more questions. Your examples for computation on a sphere are very, very convincing, on a sphere or hypersphere. But in geometry processing, and I'm sure in the real world of biology too, we often deal with more complex manifold geometry. Does GeoM stats, does that support computation on maybe arbitrary manifolds? Maybe ones defined by even triangle meshes that we love so much in graphics.
Yeah, so very, very good question. Again, it's true that I use the sphere because everybody can picture it. But yes, GeoMstats can also be used for any type of manifolds embedded in 3D first, and actually any type of manifolds defined a certain way. So if you can define a manifold, let's say one of your shapes embedded in 3D, if you can define that as an embedding, meaning a function that takes the intrinsic coordinates of the sphere of the shape and map that into the 3D world. So basically for the sphere, if you have the mapping from longitude and latitude to 3D space, and more generally, if you can give that mapping for the shape you're interested in or for the manifold you're interested in, then GeoMstacks implement what's called the pullback metric, which means that it's gonna understand the geometry of your shape by the embedding that's been prescribed. So it's taking the 3D space geometry, project that on this shape, and you have a way of computing the geodesic, which has the paths of shortened length, etc., etc. And the reason why it works for any shape or any manifold, so now I'm using shape to define the manifold itself. Mm-hmm. The reason it works for any shape is that it relies on differential geometry, which uses differential operators. So all these elements of metric, distance, geodesic, they come from differentiating some operators. And because we have these backends, AutoGrad, soon JAX, PyTorch, and TensorFlow, we can write all of these equations. And so we can get to the metric, the geodesics, and all the elements that we need. So now it's working when we provide the function that embeds your manifold into 3D space. It doesn't work for if you give it only the meshes. You'll need the function that maps each point of the mesh to the 3D space, but we're in the process of adding different ways of representing shapes and manifolds so that all of the elements can be computed from different types of inputs. But it is done from one type of input. It's theoretically possible for the other types. We are in the process of implementing that.
Thank you very much. So a few more questions for Rinat, if that's okay. The first question is, could you walk me through a little bit, now that you have the muscle geometry and you have the bones of your animal, creature, what have you, and you want to make it move, do the muscles immediately know how to contract? Is there a reasonable sort of animation that happens in the muscles that tries to reflect the bulging and the non-bulging? How does that usually work in an animation studio? Do you know?
Yeah, so that was like one of the parts that we did show some simple simulation examples, but the reviewers actually weren't too happy about them. So we kind of concentrated just on the part of generating the geometry. But where do you go from there is then you have to basically create a rig that kind of follows the skeleton mesh that you've created. So just basically bunch of joints. And then you need to figure out where the muscles actually attach to the bone by creating the constraints on the points. And the one benefit of our system is that because we use this curve representation, the curves will tell you exactly where which vertices on the muscle meshes are on each end of the muscle. And also the curves tell you the direction of the fiber field. So I didn't have time to explain it, but one of the benefits of this curve representation is that we automatically handle generation of fiber fields, and the fiber fields used in all of the physics-based simulations to basically explain in which direction does the muscle contract. So basically, I think in theory, we provide all the information needed or at least make it much more user-friendly for people who use simulation software to just kind of plug our muscles into the system and get some results. But I guess your question was, where does the animation come from? Yeah, you need the like an animator to actually start moving those joints, which would move the bones, and the bones move the muscles, which is the opposite of what happens in real life, right? Our muscles move the bones, but in visual effects and games, it's the bones that move the muscles. Not for most cases, not the other way around. Yeah.
Yeah, very interesting. So maybe I think we're a little over time here, but maybe one more question that's maybe meant more as a general discussion for both of you. I noticed that in Rinat's paper, one of the key things they do is they discretize volumetrically. Like they have this volumetric discretization where you have basically like an occupancy of muscle and bone. Whereas in some of the examples you showed, Professor, you had line segments describing your cells. Is that right? Were those actually line segments? And so I guess both of those, in both of their specific applications, I would usually think of drawing muscles with a mesh and for like animation. But then when I look at microstructures, I've seen a lot of, medical imaging papers that represented like a grid. I was wondering if you guys could talk a little bit more about the trade-offs between these types of different representations, basically an Eulerian versus Lagrangian representation. This is just meant as a broad final discussion. So whoever has anything to say can say it, and if you don't, then it's my problem. So
I can
go ahead.
Give a first answer. So actually, that's a very interesting question from a shape perspective, because what we're trying to describe, the shape, should not depend on the parametrization or the discretization that we use. So the same way the shape should not depend on translation, rotation, it should not depend on how we parameterize it. And so one element that we quotient out when we look at shape is also the parametrization. And so I have left that because I didn't want to make the conversation more complicated. But for a very simple example, with the cells that are the cell boundaries that are discretized as a set of points along the cell, the shape of the cell shouldn't depend on the way these points are sampled. And so you need to be able to quotient out the parametrization, because it's another type of confounding factor. So another question space that arises when we look at shape and when we look at biophysical information that is entailed in these shapes.
So I think because the muscles are, so when you were talking about like cells representing as line segments, I think you mentioned. So are you talking about the fibers that comprise the single muscle? Was that the
I was talking about the slide that Professor Miolane was showing, just about cells when you were looking into classifying whether a drug worked well or a drug didn't work well. Those looked like they were line segments to me. In your case, it was a volumetric. It was like more of a volumetric discretization.
Yeah, so we were kind of thinking that if you actually look at the anatomy of a muscle, each muscle consists of a bunch of fibers, like basically combined together. Fiber, I guess you can think of it as like lines. That's the best way to illustrate them. So this like curve representation is basically how can we guess overall direction and shape of these fibers within this volume, and also give kind of users control to change like the volumetric shape within the volume. But the end goal is, I guess, not to be exactly medically anatomically correct, because again, our main application is more like VFX and games. Is how can we just avoid modelers trying to guess the anatomical shape and just give you like simple tools that you can get the best approximation possible within the smallest amount of time. I guess that's the
Yes, of course. Well, thank you to both of you for your time. I really feel like I learned a lot this episode. So for the rest of the viewers, if you have any more questions, please go to the Discord. We're going to make a channel really soon to have all the leftover questions, and we'll try to bring the authors to look at that Discord channel in their free time. Apart from that, thank you very much for watching another episode of the Toronto Geometry Colloquium. Next week, we're taking a break because there's a conference deadline. There's a CVPR conference deadline. And then we're coming back the week after that. So tune in and come back to see more very fun geometry content. Thank you, everybody. Bye. Bye.